Hey guys, I'm Irene and I'll be explaining DNA's function and structure, DNA replication, and transcription and translation. So firstly, DNA and its function and structure. DNA stands for deoxyribonucleic acid, and it's a molecule that has all the genetic information an organism needs to develop, live, and reproduce. The information is inside of every cell and gets passed down from parents to their children. DNA's shape is a double helix and it's made up of molecules called nucleotides. Each nucleotide has three parts to it: a phosphate, a sugar, and a nitrogen base. There's four nitrogen bases: G for guanine, C for cytosine, A for adenine, and T for thymine. G and C are always paired together with 3 hydrogen bonds, while A and T are always paired together with 2. How these bases are ordered determines the genetic code of the DNA. Okay, so back to the structure. Nucleotides are attached to form 2 strands that spiral and form a double helix. The phosphates and the sugars are on the sides, while the bases on the strand connect with its corresponding bases on the other strand, again with G and C and A with T. Okay, now on to DNA replication. So, before cell division, DNA makes a copy of itself, and that process is DNA replication. First, this enzyme helicase breaks apart the hydrogen bonds that are holding the complementary bases together. This basically unzips the double helix of the DNA. After this, the 2 strands of DNA go off in 2 directions. Because of this, the strands are copied differently. On the leading strand, DNA replication starts with the primer, which is a short piece of RNA that's formed by the enzyme primase, and another enzyme, DNA polymerase, comes along to continuously add new nucleotides. On the lagging strand, replication is done piece by piece in the opposite direction. Primers attach themselves at multiple locations, so multiple molecules of DNA polymerase can add nucleotides to each primer at once. And last, transcription and translation. An important part of transcription and translation is RNA. RNA stands for ribonucleic acid and acts as the messenger of information from DNA. RNA also has 4 nitrogen bases: G for guanine, C for cytosine, A for adenine, and U for uracil, which replaces the thymine in DNA. Now that we're caught up with RNA, we have transcription. Transcription starts with the strand of DNA. Helicase splits the 2 strands apart, then RNA polymerase comes in to add RNA nucleotides. That one strand of messenger RNA, or mRNA, splits off from the DNA strand and goes out into the nucleus of the cell. Translation comes after, with ribosomal RNA, or rRNA, attaching itself onto a strand of mRNA. Transfer RNA, or tRNA, then comes to pair 3 nucleotides with the RNA strand as it leaves an amino acid. As rRNA and tRNA keep moving along the strand, it creates a chain of amino acids. These amino acids provide the instructions to form proteins. And all that is protein synthesis. Thanks for watching. Oh, and here's my works cited.